Chapter 6 is on pedigree analysis, applications of it, and genetic testing. So fingerprints, also known as epidermal ridges or dermatoglyphic patterns, are found on our fingers, toes, and the soles of our feet. And what's interesting about these epidermal ridges that form on the tips of our fingers is that they are believed to appear long before a fetus is actually birthed. They are thought to be formed about 17 weeks at the 17th week mark of pregnancy. And these fingerprints are permanent, they are for life. Research has shown that these patterns are and can be influenced by heredity, but there are other external factors, or factors I should say, that can alter or influence the epidermal ridges in their formation. If you were to actually look at siblings or individuals that are related, there are a greater similarity of their fingerprints than non-familial individuals. And so this has become tied to genetics because your fingerprints or the epidermal ridges that form are genetic or genetically influenced, and also are inherited. Now, why is this important, or what's the application of this outside of having these epidermal ridges? Is there is a condition known as dermatoglyphia, ADG, and this is a condition where fingerprints are completely absent, and this is an autosomal dominant trait, and that's what you see here in this picture here is a fingerprint that is superimposed on a DNA sequence. Now, what's interesting about this is because it is a rare disease, it is also dubbed the immigration delay disease, because when an individual is traveling or attempting to cross borders, they have issues with their lack of fingerprints in identification. So let's talk about, let's go into details about some of this, and not just fingerprints or epidermal ridges, but overall how certain diseases or conditions are inherited and how one can trace them. So what you're looking at here is a picture of the Swiss family, a pedigree of a Swiss family that has dermatoglyphia, the absence of fingerprints. Now let me explain a little bit more about a pedigree, because a pedigree shows the history of a family. Okay, so each, as you see here, each Roman numeral denotes a generation. Okay, and you'll see here the ones that are affected are colored in red, and the ones that are not are colored in white, but they don't show any effect of the disease or condition at hand. If there's a line or slash that goes through them, they are considered deceased. The squares represent the males, and the circles represent females. Okay, so in this specific pedigree here, those that are colored in are people that are affected with dermatoglyphia. So these individuals here suffer from the condition of not having fingerprints. So let's talk a little bit about how you can study or use pedigrees to study the genetics in humans and special features. So one of the things that are needed for this is controlled mating, which is not possible, long generation lifetime, and small family size. So it's difficult to use humans in many studies because of these features, which are special, but this also explains why pedigrees are needed when looking at or trying to understand more about a family's history. And so oftentimes what will happen is a pedigree will be used, which is a pictorial representation of a family's history for a specific trait or disease, and it pretty much acts as a family tree, and it outlines the inheritance of one or more characteristics. Okay, in a pedigree or family's history, the proband is the person from which the pedigree is initiated, so where you can trace it, the person that's initiating the need for this. So let's look at this pedigree here, specifically the characteristics or the components of a pedigree. So again, the square is the male, the circle is the female. If the sex is unknown or unspecified, it is denoted by a diamond shape. If those shapes are colored in, that denotes the person that is affected. Okay. If someone is a carrier but does not show the trait, they are going to have either a circle colored in or half colored in. Okay, I'm sorry, the asymptomatic carrier, so that means they're unaffected at the time, but they may potentially exhibit later. They may be shown with a line. If multiple persons have it, there will be a number inside. If they're deceased, a line is slashed through. If they are the one that is causing this initiation of the pedigree, they will have a P next to it for proband. If the history is not known, there will be a question mark on the inside. Now, when you want to identify the parents of or the offspring of two individuals, the parents are going to be connected by a line, as you see here, and then the line that descends from the two connected are their offspring. If someone is adopted in, they will have this denotion here, and their parents will be traced outside of that actual pedigree. These are twins, and then if you have mating between related persons, so incestual behavior, it'll be noted with these two lines here. So let's go in and look at a specific trait. In this case here, it is a pedigree of this child that has Waardenburg syndrome. Okay, and you'll notice here is the proband, so this is the individual that is initiating this pedigree. And a Waardenburg syndrome is an inherited autosomal dominant trait, and some of the symptoms of this trait are deafness, fair skin, visual impairment, and that white forelock that you see here on this little cute little kid here. So let's look at some of the things that I just talked about, the characteristics or symbols for this pedigree. So in this here we can see in generation 1, which are the parents, or in this case the great grandparents. The individual, the father is deceased, the mother is still living, and she is affected by Waardenburg syndrome. They have 2 children, okay, and these 2 children were both showing signs. That's also another clear indication that is a dominant trait. They both marry and they have children, as you can see here. 1 and 2 marry and they have 1 and 2, so 2 and 3 are their children. 4 and 5 marry or they mate and have children. 8, 9, 10, 13, and 14. Out of those children, 2 of them are affected. In this case, both are affected. Here, this individual here also had other children with another individual, and so we'll have at least 1 child with this individual. So I'm going through this because I want you to see how to read a pedigree, okay? And so each Roman numeral denotes a different generation. And when it's circled in, you can identify which ones are affected and which ones are not affected. Because it's a dominant trait, okay, you're going to see that the probability or the likelihood of is increased with an autosomal dominant trait of those offspring being affected. So for more pedigrees, there are different figures that I've denoted here that you can look in the textbook to identify all of these different traits. But I'm going to go through some, and I'm also going to give you a table and just kind of briefly give you some tips to identify pedigree analysis. For those that are going to grad school or medical school, you're going to see this come up as well. So you want to make sure that you are understanding these concepts, and it will be on the exam as well. So in this case here, you're looking at a pedigree that denotes an autosomal recessive trait. Now with the autosomal recessive trait, usually the parents don't have to be affected, but they are carriers for the trait. So as you'll see here, neither of the parents, 1 or 2, that have offspring, 3 offspring, show that they're affected with the disease, but they produce a child that is affected. And so in this case here, of parents 1 and 2, they most likely are carriers for this disease. Okay, so with autosomal recessive traits, they tend to skip a generation. And so you'll see here in generation 3, no one is showing to be affected, but generation 4, it reappears. Okay, and this is the frequency of it being affected is equal in sexes, so males and females are equal in their ability or the probability to be affected by the disease. Here you are looking at an autosomal dominant trait, and I want you to notice that in an autosomal dominant trait, Okay, at least one of the parents has to be affected. And it appears, while it appears equally in males versus females, it appears in every generation. Okay, it appears in every generation. And affected individuals have to have at least one parent that is affected by the disease, and unaffected individuals do not pass on the trait. And that is because they don't have a dominant allele. So in this case here, they do not pass on the trait because they don't have a dominant allele. An example of a autosomal dominant trait is familial hypercholesterolemia, and this is a condition where their blood cholesterol is greatly elevated, and it's due to a defect in the LDL receptor. Okay, that's the low density lipoprotein. Okay, and so this LDL particle attaches to that receptor on the cell surface, and it moves into the cell. That cell, the LDL particle is broken down, and then the cholesterol is released by the cell. And so this results in individuals that have high cholesterol, and it is, again, hereditary. Here is an example of an X-linked recessive trait, and it appears more often in males than females. Okay. However, the trait is not passed on from the father; it's passed on from the mother. So you can see here, this is the royal family, and oftentimes with things like this, the royal family are tend to experience high levels of, for example, hemophilia. So this is specifically showing hemophilia. Now, and this is classic hemophilia. The issue with hemophilia being so prominent in the royal family is because they have a lack of diversity in their mating. So royal, especially in past times, royal families were very particular in who they could mate with, and so because their gene pool was significantly skewed and reduced, it causes an influx in whatever is present there to be exacerbated. So you'll see here, Queen Victoria is a carrier for hemophilia. She has offspring, and her children Alice and Beatrice are showing being carriers, but the son is shown to be affected. Go to the next generation, you have 1, 2, 3 males that are showing hemophilia, while the others are just carriers. Okay, and you'll see that with a autosomal recessive trait, there's going to be a high prevalence of males that are affected more than females. You'll have a high level of females being carriers, but they will not show the actual trait as high or as frequent as males. There will be females that show them, but oftentimes it's every few generations. And the reason is females have to show both the mutant or the recessive form of this trait on both X chromosomes, which means the father has to have it and the mother. Whereas in males, because males only have one X chromosome, they only need to receive one recessive chromosome for the mother to express this condition. So again, with autosomal recessive traits that are X-linked, it's only showing up at a higher rate in males as opposed to females. So here we have, X-linked dominant traits. And with X-linked dominant traits, it does not skip a generation. And so what you're seeing here is because it is linked, X-linked, okay, males are going to pass that trait on to their daughters, not to their sons, because again, they're passing on the Y chromosome. However, the mother has to pass on at least one recessive or be dominant to the son. And so what you see here again is, you'll see that the affected females, if they're heterozygous, pass on the trait to about half of their sons and about half of their daughters. Okay, so in order for, in this case, an affected male, they must have an affected mother. And this is why you see here, because the mother was not affected, the males did not show it in generation 2. The only time the male showed it is in generation 3 when they had a mother that was affected. Alright, here is Y-linked traits, and in a pedigree you'll see that Y-linked traits are only going to appear in males, and they're only passed on from father to son. So you should not see females affected with Y-linked traits, because this is a father to son transmission. All male offspring are going to be affected if their father is affected. So let's look at these tables here that I mentioned to you. These tables I would strongly suggest that you learn, because these are really quick tips to identify or tools that you can apply when looking at a pedigree. Okay, so for autosomal recessive traits, again, the frequency is going to be equal to both sexes. It tends to skip a generation. Affected offspring are usually born to unaffected parents, and the frequency that you see is higher amongst incestual relationships or ancestral marriages. In dominant traits, the frequency is much higher, and it's equal among sexes, but it still tends to show up every generation, so there's no skipping of generations as we saw with the autosomal recessive. The affected offspring must have an affected parent unless they possess a new mutation, which is not very likely. When one parent is affected, the other parent is usually unaffected. So what you tend to see is half of the offspring will be affected. If the parent is unaffected, they do not pass or transmit that trait on to the offspring. For X-linked traits, you're going to have a higher proportion or higher rate of males that are going to be affected than females. The affected sons are usually born to unaffected mothers, so you will see the trait skip a generation. Half of the carriers, mothers are affected, and it's never passed on from father to son. It's always passed on from the mother, because again, the father does not have an X-linked trait to pass on to the son, only giving the Y chromosome. Any daughters that are affected is because of the transmission from the father, not solely from the mother. Because females have, or daughters have females have two X chromosomes, they have to inherit both recessive alleles from the mother and the father in order to be affected. In dominant traits, for X chromosomes or X-linked dominant traits, both male and females are usually affected. However, more females are going to be affected than males, and this is because in this case here, when it's dominant, the female can get it from the mother and the father. So there's a higher rate for the females to get this the dominant trait from the X chromosome than the son. It does not skip a generation because of that dominant trait. It does not skip a generation, and so you hopefully are seeing a pattern with dominant traits. There is no skipping of generation. It is usually appearing every generation. Y-linked traits, this is very simplistic in that it's only going to affect males, and it only can be passed on from father to son. Because it is passed on from father to son, it does not skip a generation either, because the father can only pass on its Y chromosome to the son. So every generation that there is a son, the male that is affected, they have children, and there's a male child, the male child will be affected. Okay, so when studying twins for genetic studies and inheritance, and you can see this on table 6.3, but you see that there are two types of twins, okay, monozygotic and dizygotic, okay, or dizygotic. So dizygotic would be non-identical twins, monozygotic would be identical twins, okay. With them, you have concordant traits, and that is a trait that that is shared by both members of a twin pair, and concordance is the percentage of twins that are concordant for a trait. So you can see in table 6.3, they show a study amongst twins where they were looking at obesity and the prevalence of obesity that was shown, and ultimately what they were able to identify was that for concordance, the concordance of monozygotic versus dizygotic twins, there are several traits, but you tend to see a higher prevalence in monozygotic twins as opposed to dizygotic twins. So here you'll see that A is dizygotic twins, and B is monozygotic or non-identical twins. So identical, non-identical. So you see here, okay. For the most part, overall, you have a higher prevalence of individuals that are showing these traits when they are monozygotic twins as opposed to dizygotic twins. So identical versus non-identical. Okay, they also looked at twins with asthma, and they found that there was a combination, it was caused by a combination of genetic and environmental factors. So just looking at twins to see, was it solely based off their ability to inherit or develop asthma? Was it solely based off of genes, or were there other factors? And they found that it actually was a combination of genetics and environmental factors that contributed to twins having asthma. Okay, so when they did what we call adoption studies to look at the effect of obesity on genetic influence, the question they asked was, is body mass index or BMI influenced by genetic factors? So in order to do this, they compared the BMI of adopted children with those of their adopted and biological parents. So they wanted to look at, okay, let's compare their BMIs with their biological parents as well as their adoptive parents. And the goal was to see if it was genetic factors versus environmental. So once they were planted in a different environment with their adoptive parents, did that lifestyle influence or determine whether they were going to have a higher BMI or not? So what you see here is the biological parents and their BMIs, okay? And so what they found was overweight biological parents tend to have overweight children. There was no consistent association between the weight of the children and that of their adoptive parents. So what they concluded from this study was genetic factors influence body mass index. Now what I will say is this study is a little bit skewed because it doesn't really allow them to have the same set of groups over time to evaluate this. But what we do know is environmental factors do play a major factor in this, and sometimes we call it nature versus nurture. Okay? Is it nature or is it nurture? Does nature, i.e. your genes, influence whether you are obese or have a higher BMI? Or is it nurture? Is it your environment, the food you eat, the environmental factors that you are exposed to that can alter whether you are going to have a higher BMI or not? So this brings us to genetic counseling, okay? So genetic counseling is important because it's a non-invasive way in which you can speak to a geneticist or a physician to understand the probability or the prevalence of two individuals that are planning on having children or have children that have a certain type of disease or condition that can be deleterious, okay? So it provides this understanding of hereditary conditions and how it can be applied to determining the probability of an individual having a child or offspring that is affected. So this diagram is showing you an ultrasonography that can detect genetic disorders in the fetus, and also help to locate the fetus when one wants to conduct an amniocentesis or chorionic villus sampling. So let's look into common reasons for seeking genetic counseling. Let's say an individual knows that they have a certain disease that is prevalent in their family and it can be life-threatening. Examples can be sickle cell anemia, Tay-Sachs disease, cystic fibrosis. These are just alkaptonuria. These are just some examples of diseases or conditions that are inherited, and they can have a deadly effect on the offspring. So a lot of times for example, with Tay-Sachs disease. It is a disease that is passed on between individuals that are of Jewish descent. And so oftentimes, because of the severity of the disease, parents often opt to do genetic counseling to determine the probability of them having children with Tay-Sachs, because their children die at a very young age and they suffer significantly. Okay, so that's one example. Or let's say a couple has given birth to a child prior with a certain type of genetic disease or defect, and so they want to determine, okay, what is the probability that if we have another child, they will have that same abnormality. Okay, or let's say a couple has a child who is intellectually disabled, or a close relative that is also intellectually disabled. They may want to identify what is the probability of them having a child that will have that same problem. Or we can scroll down to the 4 or 5. So let's say you have an older woman that wants to have a child, or she becomes pregnant and she wants to understand the risk of her having a child that is significantly impaired. This can be for women typically males age 35 or older. It's considered higher at risk for having a child that has Down syndrome or other types of disabilities. And so older women tend to have genetic counseling and undergo some type of post-fetal analysis for this. It can be individuals that marry within their family, so ancestral behavior, let's say first cousins, that puts them at higher risk for having a child that is potentially disabled, or some other type of condition like like we saw with the royal family, hemophilia. Okay, so there's so many different reasons why a couple may seek genetic counseling. So what are some examples outside of what I mentioned? Chromosome abnormalities, cleft lip and palate, cystic fibrosis, dwarfism, hemophilia, Tay-Sachs disease that I mentioned earlier, sickle cell, PKU, osteogenesis imperfecta. These are all examples of disorders and conditions that can be identified through certain tests to detect for whether their child is affected. And one of the ways they do this is through a more invasive method. So genetic counseling is non-invasive, and it usually starts with a pedigree analysis and a discussion. If further analysis are needed, so they identify that there are some risks that put them risk factors that are high, then the next step can be using fetal blood to obtain fetal cells to do genetic testing on the fetus. The way this is done, you can use ultrasound to monitor where the fetus is, and then they guide the needle, okay, as you see here, that is inserted through the abdominal wall into the amniotic sac to identify or to get amniotic fluid, which will contain fetal cells in them. Okay. They then take the fetal cells and culture them, and then begin to do a chemical analysis and a DNA analysis and a chromosomal analysis, which is this process is called amniocentesis. Okay. And so this allows them to collect the fetal cells and do analysis on it to determine if the child may have a genetic disease and or trait that can be life-threatening. Or you can do a chorionic villus sampling, or CVS is what it's short term for. And this is another procedure for obtaining fetal cells for genetic testing. So the difference is they go with a catheter through the vaginal area to obtain cells from the chorion, and then they're able to then do those same analyses. However, this one tends to be a little bit more invasive and can cause, if there is something that's done improperly, just like with amniocentesis, you can cause disruption of the placenta from the vaginal wall, or you can cause, in this case here, rupturing of the placenta that can result in a miscarriage. Okay, so these are some genetic conditions that are recommended for mandatory screening by the ACMG. And these are just some of these examples here. That are strongly recommended for screening. So if this runs in your family, they're most likely going to strongly recommend that you do some type of screening for it. Okay, so how are the results from the genetic test interpreted? There are so many different genetic tests that are available, and they take the results from the test to help the parents further or to individuals understand the severity of them potentially having a child that is affected. And also, there are other factors that come into play. So which we'll talk about in the upcoming chapter. There are so many factors that can contribute to whether it's different mutations or whether it's incomplete penetrance and environmental factors. And this is where you can actually have in penetrance, you could have the gene for it, but it may not show physically. Okay, so that's what incomplete penetrance is. So there's so many different factors that can contribute to whether the child will actually be affected and to what extent. For an example, neurofibromatosis is a inherited disease that results in these benign, like large masses of tumors all over the body. One could have the trait or the gene for having neurofibromatosis, but they may not have a severe form of it. Okay, so they may have to be carriers for it. They may actually be affected by it, but the effect or the extent to which they show the trait may not be as strong as someone else, right? So there's a variability in the phenotypic expression of that disease. So there are certain factors that are put into place to ensure that if someone does have a history of certain diseases, that there is no genetic discrimination and that there's a level of privacy to it. So there's many genetic tests to help with that. And this stemmed from the 1970s, where you had African Americans that were carrying the sickle cell trait, and they had a hard time finding employment and health insurance because it is costly. Oftentimes people with sickle cell anemia have what they call spasms, or they have an outbreak or an episode, as they like to call it, and it can be very deleterious where they cannot move, they have muscle spasms, they're in a severe amount of pain, and oftentimes they have to have high amounts of blood transfusion. And so they may miss work a lot. Their insurance, of course, is going to be very high, and so there was a level of discrimination against those that had diseases like sickle cell anemia or PKU or cystic fibrosis. And so there was a act that was put in place to prevent someone from utilizing their genetic information to discriminate against them for employment and for health insurance. So this is chapter 6.